Thanks, Chris. So let me get going. So what I'm going to try to do in the next 15 to 20 minutes is tell you a subset of core principles about the microbiome and give you a few vignettes on how the microbiome impacts health and disease. Bugs are all over us. They are in every body part, and they are in constant communication with barrier surfaces such as the skin, the lung, and the gut. But there's also a subset of microbes, as you'll see, that get in contact with the immune system. So before I get started with examples, a few introductions on how does one study the microbiome. And as you'll see in my next slide, it's a very complex community structure with many genes. And to do that, to study how bugs develop, how they are perturbed, and how they contribute to disease, we broadly use sort of two methods. One is called the 16S RNA gene amplification system, so to measure who's there. And then by genome sequencing, a term that I'll use often called metagenomics, we can also get to who's there and what are they doing. We can also measure the transcriptional activity of every gene in the bug, in the bacteria, and this is called metatranscriptomics. And towards the end, I'll tell you about how we can measure molecules or chemistry made by bacteria, viruses, and fungi in the gut, and that we measure by a process called metabolomics. So facts. So the human genome contains roughly about 20,000 genes that encode proteins that are defined as the units of function within an organism. And you can use that gene map to get to transcriptional events, metabolism, and many other readouts. Just by comparison, you can see the facts on this slide. They are something in the range of 100x to 1,000x more bacterial genes than the human genes in the human microbiome. And this is about how you count genes. And within these genes, they are found in something like 4,500 unique bacterial species in the gut. And the one way of looking at this is the microbial gene catalog then extends the human gene catalog and contributes to function. There are many factors that impact the microbiome in the gut and many other body sites. So the food we eat clearly alters the microbiome. The medications we take impacts it, lifestyle, and also gene content also impacts the microbiome community structure. So for example, if one of us go on a gluten-free diet, you can see how this particular taxa changes in abundance when you switch from a regular diet, or what I refer to as a habitual diet, to a gluten-free diet. So a single intervention here changes an important microbial community component of the microbiome. Bugs do many things. They also modify dietary compounds, and I'll given you a few examples here. They also decrease or increase the bioavailability of some of the drugs we take. And then, as you'll see, they modify many of the host-derived metabolites. So they could be bile acids, cholesterol. I'll give you an example of that. And the bacterial effect on these metabolites alters the function of these metabolites, both in good and harmful ways. The second concept that I'd like to highlight is that the microbiome measured by what we refer to as a diversity measurement changes as we age. And these phenotypes can be measured over time using the two methods that I described a few minutes ago. So a few years ago, we collaborated with a lab, Kenya Honda's lab in Japan, where they recruited almost 319 Japanese individuals, and you can see the breakdown of this age group. So 100 or so that were over 100 years old, an older group, and a young group. And majority of these individuals who were recruited this study were free of chronic disease. So the impact of disease on the microbiome was minimal. And what was striking is what I'm showing you is that I mentioned to you this component of bile acids, which is a host-derived set of molecules, got modified in centenarians compared to young individuals and older individuals. So the clear effects of gut bacteria on secondary bile acids. So what does this mean? So two observations. So this conversion of bacteria on secondary bile acids increased the ability to fight pathogens. So the centenarians who had this change in chemical structure were more resistant to infectious disease. And these same compounds also altered the balance of T cells. So T cells that are regulatory in nature or inflammatory in nature. And these compounds altered that balance. So again, finding many ways of keeping harmful pathogens away from us. And the change in the T cell phenotype impacted our susceptibility to inflammation. A second vignette. So this was an example where we were lucky again to study children born in Finland, Estonia, and the border towns of Russia. And the reason we wanted to study this group was, as you can see from this slide, the incidence of type 1 diabetes in kids born in Finland sky high. So could we use a living laboratory to identify whether there were external factors, in this case, the microbiome, and the risk of disease? All these individuals had the same risk of type 1 diabetes. So they were HLA genotyped to be sort of a common starting genetic risk. And the studies were done when these children were mostly breastfed. So their diet, again, was very similar if you happen to be born in Finland versus Russia. So what did we learn? So if you look at this slide, it's a little complicated, but what we could see, if you look at the first year of life, there was a certain bacterial structure that was higher in kids born in Finland compared to the kids born in Russia. Without going into all the details, if you look to see what products these change in bacterial community structure contributed to, was a product called LPS. So this is a common bacterial membrane that is recognized by the immune system. And the LPS that was predominantly made by kids born in Russia was an LPS that came from E. coli, whereas the children born in Finland or in Estonia, the LPS came from these species called Bacteroidetes. Now that LPS, when we looked at the structure, had key differences. So normally, the LPS made by E. coli has a 6 acyl chain LPS, so these 6 extensions, whereas the LPS made in Finland had 4 and 5 acyl chain LPSs. And what we were then able to show is that if you fed mice who had risk for type 1 diabetes, their acceleration of disease depended on the type of LPS that they were exposed to. And when we went back and looked at the kids, what we learned was being exposed to E. coli-derived LPS in the first year of life, so those are children primarily born in Russia, their immune system was educated. So they had the ability to reduce the risk of type 1 diabetes and other autoimmune diseases, whereas the kids born in Finland did not receive that education signal, and their risk of autoimmune disease was significantly higher, and in this particular case, type 1 diabetes. So in the second example, I'm highlighting differences in the gut bacteria may affect immune maturation and your predisposition to immune-mediated disease. So this early life exposure is again an important signal for immunity later in life. So I've given you 2 examples. I've told you about centenarians being protected from inflammatory disease and various infections because they modified bile acids. Bacteria within them modified bile acids, and this bile acids had protective effects on the centenarian population. And in this second vignette, I've told you about the risk of type 1 diabetes. So the question that one could ask is, are we losing our friends as we westernize diet, and our environment. So I've told you so far that microbial exposure early in life is important for immune education and susceptibility to autoimmune disease. And in the second vignette, I've told you about centenarians having a distinct microbiome and higher levels of this special class of secondary bile acids that's associated with antimicrobial effects and anti-inflammatory effects. So the third example is, I think I briefly alluded to in one of my earlier slides, that cholesterol biosynthesis goes through some defined steps. And many of the enzymes that mediate this cholesterol biosynthesis have been discovered. The discovery of HMG-CoA reductase led to a Nobel Prize, and the discovery of the commonly used statins. And so one of the students who joined my lab as a graduate student, Doug Kenny, a super smart guy, was interested in this observation. So in the 1930s, there was this observation that something in stool samples could convert cholesterol to coprestanol. And over the series of almost 100 years, there have been many attempts to try to identify the enzymes within bacteria that bring about this conversion. And so why is this conversion to cholesterol to coprestanol important? As you can see here, as cholesterol comes from diet and bile, and the microbial conversion of cholesterol to coprestanol basically is excreted. So coprestanol is not reabsorbed back into our bodies. So if we keep doing this, we can excrete a large amount of cholesterol converted to coprestanol daily in our stool, suggesting that this might be another mechanism to keep serum cholesterol levels lower. So what Doug Kenny did during his graduate studies asked what genes are responsible for this conversion, which bacteria have these genes so that they can do the reaction, and what are the consequences of intestinal cholesterol metabolism, and how does it affect serum cholesterol levels. And I'll go through what he did. So he first came up with an assay to be able to take a stool sample and measure how much cholesterol and coprestanol is in the stool sample. So you can see that. And then by looking at a large population structure that included individuals from the Framingham Heart Study, from individuals born in the Netherlands and also in China, he was able to look at this metagenomic data that I told you about to try to identify what we call encoders or enzymes that can convert cholesterol to coprestanol and those who didn't have this enzyme activity. And I'm skipping through a lot of detail, but what he was able to identify was initially starting with this bacteria that previously other investigators had shown had some activity in this conversion reaction. Doug was able to identify this enzyme that he called ECOP170 at that time that could bring about the initial conversion of cholesterol to intermediates that end up as coprestanol. And so by doing some clever biochemistry, he was able to identify the protein sequence that was required for its enzyme activity, and then go on to show that the presence of this enzyme activity could convert cholesterol to coprestanol, and the gut could function as a sink, removing cholesterol in the form of coprestanol. So what I've told you is, again, one vignette of how we've been able to identify another way of decreasing total cholesterol levels in our blood by sort of excreting it in our stool, and offering the potential that with additional work, one might be able to find a combination of enzymes that could be used to deplete serum cholesterol levels in addition to the currently prescribed drugs. This conversion was quite powerful when compared to the currently prescribed drugs. So in the third vignette, I've told you that as an example, I've used microbial cholesterol metabolism as an example. And so you can begin to dream about identifying many more of these enzymes that is going to have potent activity on human health and human disease. And by carefully studying microbial function, today I'm mostly focusing on bacteria, but there are similar contributions from gut fungi as well as gut viruses, but that will be another day's discussion. And we're now beginning to learn that the gut microbiome has much broader effects. So the effects that I've described in the previous 3 vignettes are effects that are local, so the effects that are in the gut. But now emerging evidence from many groups are beginning to show that broader phenotypes such as mood, depression, anxiety, sleep may have important contributions from gut bacteria and the products from these gut bacteria. So without going into detail, there's a long list of diseases, including food allergy that's affected by the microbiome. And one of the goals of the Food Allergy Science Initiative is to try to identify these bacteria, find the function of these individual bacteria, how they might impact various food substances or the immune system, and increase the risk of various allergies to food. And I'm confident that over the next few years, we'll make significant progress in this on this topic. So to end, I'm going to tell you about how might one start thinking about therapies with the microbiome. So one could use the bacteria themselves as therapies. So this is commonly referred to as microbiome transplant experiments. So it could be the entire microbiome of an individual, or you could select a subset of bacteria that could be used to correct the missing bacteria. You could also measure the products from these bacteria, and these would be referred to as microbial metabolites, or the antigens that then impact the immune system, as I showed you briefly in the type 1 diabetes example. So the sort of the standard example was this disease called Clostridium difficile infection. So this is a diarrhea type illness that you get when you are exposed to antibiotics. And as you can see, the toxin that's made by this bacteria contributes to this disease. I mean, in a landmark study that was published a few years ago, almost 10 years ago, the individuals who had this diagnosis were treated with the standard of care antibiotics, and a subgroup of individuals were given this microbiome transplant experiment. And what was striking was you could clearly see how the infusion of this microbial community corrected the microbial community structure in these kids and adults who had C. difficile infection. But the really other striking observation was that this transplant allowed you to get back to a healthy status. And last month, the Food and Drug Administration approved the first such treatment in the form of a capsule for patients with C. difficile colitis. And my expectation is that there would be many additional studies. Having said that, the early success of putting back the missing microbiome has not worked for every disease. And so we in the lab have been interested in finding out, could we find products made by the bacteria, so chemical compounds that could serve as early drugs to correct the missing community structure, or could we then also start learning about the immune system? So here's an example where we did an analysis where we looked at something like 500 patients with inflammatory bowel disease and looked at their microbiome, so roughly 2,000 samples from these individuals. And so we can now begin to say, okay, what are the bugs that increase in inflammatory bowel disease and what are the bugs that decrease? And I'm just giving you a snapshot of this data. So we found that Ruminococcus narvis was increased in patients with inflammatory bowel disease. And so we basically set up a very simple assay. Could we find the molecule made by Ruminococcus narvis that impacted the immune system? And if we could do that, then one could start thinking about treatment. Or at least in this case, why do patients develop inflammatory bowel disease? And I'm capturing this. So what we do is we take the bugs that come from these individuals, we break down the bug into various fractions, put them on immune cells, and ask whether they make certain cytokines. So do they make cytokines that are inflammatory in nature, so something like TNF alpha, or anti-inflammatory in nature like IL-10? And we then try to find out what is the chemical structure that brings about that reaction. And in this particular example, we were able to identify the molecule made by Ruminococcus narvis and go on to show that this signal affected or increased the risk of inflammation. So in the last vignette, I've told you about how a particular bacteria associated with disease can make the disease worse. And in the final example, we can also look at antigens that are present in this bacteria that are recognized by human T cells, and use that information to sort of find why patients are at high risk for various autoimmune diseases. So the approach we took here was same, starting with metagenomic data. We came up with a computational algorithm to predict these antigens. We synthesized these bacterial antigens and looked to see how they may impact T cells in this case. And what we've been able to do is, using a series of assays, try to get to this observation that certain epitopes or antigens made by healthy individuals, the microbiome in healthy individuals, when they see T cells make a cytokine that's good for you, something called IL-10, whereas if you take the T cells from children with Crohn's disease, they make cytokines that are harmful and accelerate inflammation. So what I've told you today in the last 18 minutes or so is to tell you briefly, starting with what's the microbiome structure in all of us, what are some of the key features, how do we study these bacteria, and then I've given you a few vignettes to tell you about how microbiome impacts the risk of autoimmune disease, how it may maintain health in centenarians, and so something that we could build on, and then given you 3 examples of how the effects of bacteria on cholesterol, and one might be starting of a therapy for these for people with high cholesterol levels, and then told you also about how do bacteria contribute to disease. And my hope is that going forward, we'll be able to, we've already started some of these studies to figure out how does this work in kids and adults with various food allergies, and using this blueprint that I've discussed today, I'm very hopeful that we'll make rapid progress. I'll stop there and I'll be happy to take questions.
That was excellent, Ramnik. Thank you. It's just amazing to think about the microbiome and how far we've come, yet we still have a long way to go. I know when I was in medical school many, many years ago, this was not even discussed at the time. So it's pretty amazing that we're seeing it now in many different diseases. And what I'm really excited about is how you're taking all your experience in research in IBD and able to apply that to the food allergies as well, which is great for our food allergy community. So we'll know more about that. But as you think about this, one question that always comes up is, the microbiome is vast. There's so many different bugs. We don't know how they all function. And you go to the local markets and there's a million different probiotic products there. And how do you, with your patients, kind of guide them besides, stay away from them? What do you do to guide them in that model?
Yeah, so the probiotics, most of them were developed in the pre-metagenomic era. So before we had lots of information about the complexity of the microbiome and the genes within the particular bacteria. And you saw in that example that I gave you for C. difficile colitis, when you have a gut that is significantly depleted of bacteria, then it's a lot easier to put bacteria back in. But what we and others have learned is when you have a gut that is filled with bacteria, many of these probiotics only stay around for a short time. As I alluded to, what's urgently needed is to come up with quantitative readouts and not subjective readouts, where one takes a probiotic and says, okay, I feel well. But I think what I was hoping to get through today is that figuring out exactly how the microbial gene might work or the microbial product might work is where I think we're going to have a huge impact. Now, with a disease like food allergy, most of the activity is in the upper part of the gut. And so one needs to focus in on bacteria that are found in the upper part of the gut and readouts that are relevant to triggers of food allergy. Now, it's also possible that these bacterial enzymes could modify some of the foods that are associated with an increase in allergic symptoms. And we also want to find how does this happen, and could we then find ways to prevent it?
Yeah, and as some people might not know, that the upper gut is a little bit harder to analyze because you just can't so easily take a stool sample and see the end result. You have to go into that area of the gut and take a look in the sample there as well. Probably the best way to go, which we are doing a clinical trial on that right now. So hopefully we'll get some interesting information from that. And then let me just look at the questions here. It seems like industry and investment community are still apprehensive to develop microbiome-based drugs. Why do you think that is? And do you think a need for the industry to start pursuing microbiome-based drugs more aggressively?
Yeah, so there was a lot of excitement when the fecal microbiome transplant experiments worked in individuals with C. difficile infection. And so there was a lot of excitement, and everybody tried to come up with a similar approach to treat type 1 diabetes and obesity. And, you can think of that long list that I had on one slide. Mm-hmm. But the success there has been limited. And I think what industry and academia need to do is to figure out the underlying mechanism. So how does the microbiome impact disease? What are the triggers that the microbiome contributes to or attenuates? And the approach we've taken is we've gone away from bugs as drugs, and we'd like to explore the option that bugs make chemistry or drug-like molecules, and if we can use that type of information to design better molecules, that might be a more fruitful approach to treating diseases. So I think staying on the outside, industry is asking for more information so that they can better design the studies within large and small pharma to sort of get closer to mechanism-based drug discovery.
Yeah, and that's critical. And this is why FOSSI was started, right? To understand the biology of the disease. And that's the only way to move forward. And then the other question, when you think about what you were showing with, the type 1 diabetes and the differences of increase in certain Treg cells versus inflammatory cells, do you think there's a way, so let's say you know that, how do you switch that? Is there a way to switch it back?
So the
molecule, or where has that gone?
So the
same in IBD actually as well.
Yeah, so the early attempts are promising, so that if you can find these key triggers, then one can. There are many ways of doing this. So one is you could have a biologic that's prescribed for a disease. And generally, this biologic works well, but with time, patients develop resistance. And the resistance could be many factors, but one component is the microbiome. So that if we could identify the triggers or the culprits behind resistance, then a treatment strategy could be done much earlier. So when the patient is responding to the biologic, you use microbiome-based therapies to prevent or delay the emergence of resistance. The other potential example could be there's some early evidence that some of the drugs that are prescribed for mood disorders can be metabolized by microbes in us. So that decreases the bioavailability of that drug. And so if you could find ways to interfere with that process, the possibility is that the drug levels would stay high and patients would benefit from those drugs. And what we sort of inactivate or turn off is a small set of enzymes in the bugs, not the entire bacterial community.
Yeah, there's interesting data in oncology as well showing metabolism of drugs with PD-1 inhibitors and benefit with certain microbes over others. So it's quite the field. Here's another question. Dear Professor, do you think it's a better way to recognize the gut microbiota as an ecosystem and analyze it as a whole instead of looking to one bug at a time?
Yeah, so as you have noticed, all our experiments start with the ecosystem. And then the ecosystem gives us patterns of change. And then you use that type of information from a large number of individuals to become more precise. And by doing that, we have the opportunity of sort of starting with lots of data, but then having a disease-related hypothesis and then narrowing down our answer to try to prevent, correct, or treat what may have changed. Because what we learned from fecal microbiome transplants, it works very well with C. difficile, but many of the other conditions, the success rate has been limited.
Yeah, and I always wonder around that, is there an influence of diet and environment that it just doesn't last as well on there? And that's hard to figure out as well. Okay. Well, thank you so much for joining us today. We're grateful to have you as one of our investigators, and your research is critical to us moving forward. FOSSI has single-handedly transformed how we diagnose and treat food allergies, and we will not stop until we find the cure. Scientists and advocacy groups all over the world are watching our cutting-edge work, which is solely funded by generous donors. We need you to help power our next breakthrough. Donate now at foodallergyscience.org. 100% of your donation goes directly to funding scientific research for a cure. And we hope to see you next month at our next webinar. Thank you. Thanks so much, Dr. Xavier.
Yeah, thanks.
Bye. Excellent talk.